Welcome to Chapter 2, Chemical Basis of Life. So this chapter deals with, obviously, chemistry, and it deals with small things, tiny particles, all the way up to molecules. So let's jump in. This first question is asking to define different words. And the first one is matter. Now, matter is a really general term. Matter is pretty much anything that occupies space and has something called mass. All right, and then I'm going to define mass. The mass is the amount of matter in an object, and I know we're kind of going back and forth, but it's just kind of the amount of stuff in an object. And if there's stuff and it takes up space, then we call it matter. So let's go ahead and put that in here. All right. Now weight. Weight is interesting. So weight is the effect of gravity on an object with a certain mass. All right. So what that means is the weight of something can be different depending on, let's say, what planet we're on or where there's, different gravitational forces. So here on Earth, the gravity acts on a specific mass, okay, an object that has mass, and then it gives you a certain weight. Does that make sense? So if we're on a different planet with a different gravitational force, that weight will be different. Okay. That leads us to an element. All right, so now if we take matter and we look at it its simplest form, where the simplest form is only one type of atom, not necessarily one atom, but only one type of atom, we call that an element. And when we look in like chemistry, there's a periodic table that represents all the elements that are found. So again, an element is the simplest form of matter. It has unique chemical properties, and it's made up of only one type of atom. All right. The last thing is an atom. So an atom is the smallest particle of an element. So as I mentioned, that an element can have more than one atom, but all the atoms are the same. Okay. So let's take a look. Give you an example. Diagram. All right, so here are some common atoms found in living things. Okay, there's a hydrogen atom, carbon atom, oxygen atom. Now, the oxygen that we breathe isn't this. It comes as O2. So that's two oxygen atoms together. Carbon, we can put carbon together in a certain way and get like, what is it, graphite, I think, or not graphite, but like, to write, like the lead in a pencil. Oops, sorry, am I sharing again? Okay, but if we look at like, right, so I was talking about carbon, so we can put a whole bunch of carbon atoms together and form graphite. So graphite is used like in pencils, and so it's still its elemental form, but it's only made up of carbon atoms. Okay, I hope that helps you to understand the difference between those. So we can see an atom here. There's different atoms: hydrogen, carbon, oxygen atom. There's different parts to that atom. The atom, there's an atomic nucleus. And I distinguish atomic nucleus from like cell nucleus, or just what we would consider a nucleus. In a cell, we'll look at that in the next chapter. There's, DNA and things like that in a nucleus. It's an organelle in a cell. But here it represents kind of the center of the atom. And so we call it an atomic nucleus. All right, just to distinguish that. So obviously different types of atoms, and they have different properties. Okay. Inside the atomic nucleus, there's certain subatomic particles, and then kind of floating around or whizzing around the outside of the nucleus, the atomic nucleus, are other subatomic particles, which we'll look at next. All right. Okay, so now let's take a look at some of these subatomic particles. The first one is called a proton. Proton. All right, let's take a closer look. A proton, is a particle, subatomic particle, with a positive charge located in the atomic nucleus. So if we come back here, notice the P's, which represents proton, is in the atomic nucleus, all right? And it's got a positive charge. Okay, so there's an overall charge to it of a +1 charge. A neutron is a particle with a neutral charge, meaning it doesn't have a positive or a negative, and it's located in also the atomic nucleus. So inside you have neutrons and protons. The electrons, which is the last type of subatomic particle, it has a negative charge, and it's located outside the atomic nucleus. So it kind of orbits the atomic nucleus. What keeps it from like flying off? It's the positive charges on the inside. So there's positive charges on the inside. The negative electrons are on the outside. And what attracts it are those opposite charges, right? Opposite attracts. So positive in the nucleus, negative on the outside of the electrons, and they're held together in that way. But the further out you go, those negative charges are held onto the nucleus less strong, okay? So they're more likely to fly off or to be, utilized in some other way. And that's important. All right, let's take a look at something called an ion. So let's define the ion. So if we take an atom, which is typically overall no charge, right? Because what we learn, and I'm not gonna, if you take chemistry, but what you learn is that the protons and electrons are balanced. So in carbon, for example, what distinguishes it from oxygen is the amount of protons, neutrons, and electrons. So in a carbon atom, there's 6 protons, 6 neutrons, and 6 electrons. Okay, so they match each other. In an oxygen, it's 8 protons, 8 neutrons, and 8 electrons. Okay. Now, kind of an exception is hydrogen. A hydrogen atom doesn't have any neutrons. There's 1 proton in the nucleus and then 1 electron around it. But yet it's still, the overall charge is 0 because they cancel each other out, right? You have 6 positives, 6 negatives, and they cancel each other out. And so there's no overall charge. All right, an ion is an atom. So you take one of those atoms that either gains or loses an electron. So those electrons on the outside can be interactive with other atoms. Okay, there's different forces that can either, cause an atom to lose an electron or to gain an electron. All right, but whether it has gained or lost an electron, we now call it an ion. Ions are charged atoms. Okay, so if it gains an electron, it will have an overall negative charge. If it loses an electron, it will have an overall positive charge, right? So when we look go back here. All right, let's see our carbon here. There's 6 positive charges, protons, and then 6 electrons, negative charges. So if we lose an electron, one leaves, there's going to be 6 protons and only 5 electrons. So we have an overall +1 or positive charge. If we gain an electron, let's say this one gave one up and it came over to hang out on the carbon, okay, now we have 6 protons and 7 electrons. So now we're going to have an overall negative charge, -1 charge. All right, so an ion is gaining or losing an electron. All right, now we're going to build, right? So we have atoms. How do we make a molecule? How do we put those atoms together to form molecules? And so these connections between atoms are called bonds. So bonds hold atoms together to form molecules. Let's say this, together to form molecules. Okay, now there's different types of bonds. So let's take a look at a different type of bond. The first one is called an ionic bond. So obviously, just from the name, it is a bond formed between ions. Okay, I'm going to give you an example. An example would be table salt. Table salt. All right, so I'm going to show you a picture. So table salt is made up of 2 different atoms, a sodium atom and a chlorine atom. Okay, but actually it's a sodium ion and a chlorine ion. All right, so maybe I should put here in parentheses that Na is sodium, and it has to do with, oh my God, it has to do with, because you think, why is it Na and not So or something like that? It comes from a different language, natrium, I'm not sure if it's like Latin or Greek. Natrium means sodium, so that's why it comes from. Chlorine, all right, is Cl. So if we take an ion, or let's first take our atoms, a sodium atom and a chlorine atom. Now sodium has, let's make this a little bigger. Sodium has 11, I believe, protons in the nucleus and 11 electrons. 1, 2, 2, 4, 6, 8, 10, 11, right? All right, so the thing I didn't mention before is that electrons fill different energy levels. And so the first energy level closest to the nucleus, it takes a maximum of 2 electrons to fill it. Okay, so once you have 2 electrons, it fills that first level. Then any extra electrons that that atom might have goes into the next energy level. All right, and so the next energy level holds a maximum of 8 electrons. And so in this case, with 11 electrons, we can fill the first energy level and the second one also, but then we have 1 extra electron because there's 11. And so this one sits in the very next energy level. And I think it's also 8 that makes it complete. And an atom wants to be stable. So stability is reached by filling those outer shells. Okay, so the more filled those shells are, the more stable it is. If it is not filled, it becomes less stable. Okay, and I just think of that lone electron sitting out here in the sodium atom. It's not very happy out there all by itself. So it's very prone to being lost to something else. And in this case, a chlorine atom here with 17 electrons, okay, Let me. All right, so we have this chlorine atom, which is almost filled. This outer shell is almost filled, but it has one opening. All right, see there's a little open dot. So it's got 7 in its outer shell. So we can kind of see what's going to happen. We have this unstable sodium atom. It wants to lose that extra electron. And here we have a chlorine atom, which also wants to gain an electron to complete its outer shell. And so that's what happens. But we end up getting ions instead. So in order to be stable, now this sodium, okay, while stable, it's an ion because it lost an electron. All right, so it's got a +1 overall charge, whereas the chlorine atom, to be stable, it gained an electron, which gives it an overall -1 charge. And because of that, those two atoms or those two ions are now attracted to each other because one's negative and one's positive. And so that is what holds those two ions together. This is called an ionic bond. It's a bond between ions. All right, and table salt is an example of an ionic bond. All right, the next type of bond, that's one type of bond. There's different kinds of bonds. I'm going to just show you 2. The next type of bond is called a covalent bond. Okay, let me see. It should be okay, a covalent bond. I look at the word co, like if you cohabitate with someone, you share living quarters. Okay, if you are codependent, you depend on another person. You share dependency, I guess. What's another word with cooperate, right? You share tasks or so co is that term means sharing. So a covalent bond is a connection in which 2 atoms share electrons. Okay, because sometimes an atom doesn't want to actually give up its electron. Instead, it will share it with another atom. So let me give you an example here. Oops, I'll put it in place. Oops. All right, let's back in. Connection between atoms that share electrons. All right. So an example I'll give you is hydrogen. Hydrogen comes as 2 hydrogens that share an electron. Okay, so I mentioned before that hydrogen only has 1 proton, 1 electron, and we know that we want to fill that outer level. So since it's only 1 electron, it doesn't quite fill that first level, energy level. So what does it want to do? It wants to fill it. So it takes and borrows, shares an electron from another hydrogen atom, and together that those 2 electrons spend time on both of those atoms. And then that, molecule now, right? We're building molecules. NaCl is a molecule of salt, and then H2 is a molecule of hydrogen. All right, and this, of course, is a gas. All right, so there we go. 2 different types of bonds. There's other types of bonds, and you can read about them, but those are the ones that I will test you on. All right, let's keep going. So now we have, the ability to make molecules. What are some types of reactions that can occur in our body? All right, we process food, we break down things, we build up things, we get a cut, we have to heal, we have to build molecules and build more tissue. All right, so let's take a look at a chemical reaction. A chemical reaction is the ability to either build something or break it down, okay? And this occurs between atoms. So kind of either building molecules or breaking down molecules, and or making ions or whatever is a chemical reaction. And there's lots of chemical reactions in our body. I'm going to give you some examples of specific types of reactions. One is called an anabolic reaction. Okay, anabolism is a synthesis reaction. To synthesize something is to build it. Okay, which means we take smaller atoms or smaller molecules, and we put them together and we make something bigger. Okay, the opposite of that is catabolic reactions. So this is what we call a decomposition reaction. When something decomposes, it falls apart, comes apart. So in this reaction, we're breaking apart something bigger into smaller units. All right, and so that's how, our body all day long, all the time is building and breaking down, right, to heal, to make things. So if you're working out, right, you're building muscle, all right, those are anabolic reactions. So also if you take like an anabolic steroid, anabolic or anabolism refers to synthesizing or building. All right. Let's define some important terms in terms of, humans. So let me bring up some of these. So pH. So you've probably all heard about, pH, the pH of something, the pH of water, the pH of your shampoo. What is that? pH is, it stands for power of hydrogen. So we're talking about hydrogen ions. The power of hydrogen, or the, another term, instead of power, it's, I can't remember, but it deals with hydrogen ions. Okay, so this is an H plus ion. So it lost its electron, it's one electron, and now it has an overall positive charge. Hydrogen ions are very important in our body, and it's important that we regulate hydrogen ions. Hydrogen ions are very, what we call, reactive. They can be added to things very easily, or pulled off something very easily. So it's important that we keep track of our hydrogen ions, and pH is a measure of the amount of hydrogen ions in a solution. Now, a solution is just a, usually something is dissolved into something else. So maybe I should put that here. A solution is made up of a solute. Solute is what we're dissolving, and it's dissolved into a solvent. A lot of times the solvent is water. Okay, so solvent is the stuff that you're dissolving the solute in. So if we're making sugar water, for example, the solvent is water and the solute is sugar. And so we dissolve it in there and we get a solution. All right. So pH is a measure of the hydrogen ion concentration in a solution. So terms that we use in terms of pH, one is acid and one is base. Let's take a look at what those are. Because, you've probably all heard these terms, but what does it mean? An acid is a proton or hydrogen ion donor, okay? So it gives up hydrogen ions. So if you take something and you put it in water and it gives off the hydrogen ions, it's an acid. Okay, whereas a base is a proton or hydrogen ion acceptor. In other words, when you put it in water, it binds to hydrogen ions. Okay. And if we look at a table, of pH, things that have common pHs. We're going to also define what a buffer is. Okay, so I'm going to put that here, and then we're going to answer another question about why is it important to regulate our pH. All right, and so let me add a picture here. Okay, make it bigger. This is a pH scale, kind of some common household things and what kind of pH it has, what its concentration of hydrogen ions, pretty much. So we start with water. So water has a pH of 7. Okay, so 7 is what we call neutral. There's an equal amount of hydrogen ions and what we call hydroxide ions. So oftentimes an acceptor will have an OH on it. So in this case, water, which is H2O, okay, H2O, 2 hydrogens and 1 oxygen, has an equal amount of hydrogen ions and OHs, hydroxide ions. So that, and they cancel each other out. So hydroxide is negative. And so maybe H2O is made up of hydrogen ion plus an hydroxide ion, which is negatively charged. So you put those together and it's neutral. You don't get an overall charge, but it's equal amounts of hydrogen ions and hydroxide, so it is neutral. It's 7. As we add hydrogen ions, okay, as we add hydrogen ions here, the pH drops. So a low number hydrogen pH, a low number pH equals more hydrogen ions. It's what we call an inverse relationship. Okay, so if something increases, something else decreases. Right, so in this case, if you remember, pH and hydrogen ion concentration are inversely related. So the more hydrogen ions you get, the lower the pH number. Okay. On the other hand, the higher the pH means lower the hydrogen ion content. Okay, so a pH of 14, sodium hydroxide. Okay, it has a very high pH but a very low hydrogen ion content. Okay, versus when we get down here to stomach acid, hydrochloric acid, has a pH of 1 or less than 1, and it has a very high hydrogen ion content. All right, so this is, like if we wanted to know the amount of hydrogen ions, concentration of hydrogen ions, you can see how the higher the pH, the lower the hydrogen ion content versus the other way around. Okay. A buffer. So buffers are special molecules that help us in our blood and other parts of our body, but resist changes in pH. Because again, we don't want our pHs to fluctuate. We don't want our hydrogen ion contents to fluctuate a lot because again, those hydrogen ions can be very reactive. And so we have molecules that are floating around that bind to excess hydrogen ions and then give up excess hydrogen ions when we need it. All right, so when we have too many, it adds it to itself. And then when we don't have enough, it gives it up. And so bicarbonate ion is an example of a buffer in the blood. So this is an example. All right. Oops, let me make that smaller. You can make it bigger if you want, but I want to fit it in. Okay, so oops. Get a little go. Oops. Oops. Okay. Trying to move this in the right place. All right, it's supposed to be a little arrow. Okay, maybe that's as good as it gets. Okay, so we have an arrow because it's a reaction. So we take bicarbonate ion, and if there's too many hydrogen ions in the solution, then it can add to the hydrogen ion, and we can all right. And yeah. Oh, this is hard, isn't it? All right, how's that look? All right, kind of. It's going both ways. All right, and then we can take the hydrogen ion out of the blood because it's getting too acidic, and it incorporates it into a carbonic acid molecule. And then on the other hand, if we don't have enough hydrogen ions in our blood, it can go the other way. Right? We can release the hydrogen ions and get bicarbonate ions again. Mm-hmm. All right, and the last question was, why do we need it? Why is this important? Because great fluctuations in pH result in death. Okay, so we don't want to die. Oh yeah. Oh, okay, I know why. Dang. Okay. All right, so that's that question. Let me see if I can take this whole thing, and of course it doesn't. So I gotta move this one, move these little arrows again. Okay, you guys can mess with it. All right, anyway. So that's that. Let's move on to the next one. Now we're going to start looking at some molecules. Yay. All right, so we're building now. We built, we have atoms now, we have ions, and now let's put them together to form some important molecules. And I call these biological or organic molecules. The term organic in chemistry just means carbon containing. It contains carbon in it. Biological refers to being part of living things, right? So carbohydrates, lipids, proteins, nucleic acids are all types of organic molecules. Okay, a little bit about them. Carbohydrates are sugars, starches, lipids, fats, oils. We'll talk about phospholipids and proteins, enzymes, and hormones. We do spend some time on hormones, especially. And nucleic acid is DNA. And RNA, you might not have heard of RNA before. All right. How do we distinguish organic versus inorganic? So I've already mentioned that organic refers to carbon containing. So if it doesn't contain carbon, then we can call it an inorganic molecule. So, you know, when you take chemistry, you can take organic chemistry or inorganic chemistry. All right. So inorganic would be, you know, things that don't have carbon in it. All right. Let's start with a carbohydrate. And we're just going to go through the different types of organic molecules and talk a little bit about each one. So the first one is, what are some different types of carbohydrates and what do they do? So carbohydrates oops. Carbohydrates as listed up here. Oh, sugars, starches, and maybe I need to put the function. Major roles. Carbohydrates are our main fuel. They act as fuel, okay, for running different chemical reactions. And maybe we'll have time to talk about some of these reactions. Okay. It can be structural. Okay, they can be used to make up parts of our body. We need carbohydrates, let's say, for receptors in our body that receive molecules. And then the third one is that they provide bulk in our digestive system. Okay, so in the book, I said it's used as fuel. Maybe I should do this way. So the first one is energy or fuel. Or provides energy. Or fuel for the body. All right. It's structural in that it is a part of a structure of molecules. And then lastly, it provides bulk and digestion. And, so we can move digestive contents along and then also make feces. All right, so the different types of carbohydrates are the monosaccharides, disaccharides, and polysaccharides. Mono means one. Saccharide refers to sugar unit. So a monosaccharide is a simple sugar, a single sugar unit. And we can put sugar units together to form larger molecules. So I call it a subunit for carbohydrates. It's kind of the basic unit of a carbohydrate is a monosaccharide. So an example is glucose, C6H12O6. There's 6 carbons, 12 hydrogens, and 6 oxygens in this particular molecule, but it's still considered a small or a simple sugar. Glucose is really important. It's one of the main fuels in our body to make ATP or make energy. Disaccharide, it's 2 sugar units. And for example, table sugar is an example of a disaccharide. It's made up of one glucose unit and then another unit called fructose. It's another simple sugar. All right, so you put them together and you get a disaccharide. Now, what do you think a polysaccharide would be? Poly means many. So polysaccharide has many sugar units, and here's some examples of some polysaccharides. Starch, okay, it's a energy storage in plants. So plants, when they undergo photosynthesis, their end result is glucose. They make glucose, but if they make more glucose than they need, then that glucose excess is stored in the plant in the form of starch. So, we think of rice and other things that are starchy material. Well, that starch is just a whole bunch of glucose units put together and stored inside the plant. Glycogen, on the other hand, similar in its function as starch to plants. Glycogen is our fuel storage. So when we get excess glucose, all right, it's put into, put together inside, let's say, our liver and forms something called glycogen. So glycogen is just stored glucose in our body. And when we need more glucose, so for example, if you haven't eaten for a long time and you're starting to feel weak, okay, the stored glycogen in our liver is released, and the glucose units are released into the blood, and then we can utilize that glucose as fuel. All right, and a third type, Oops. A third type of polysaccharide is cellulose. Cellulose is a component of plant cell walls. Okay, and so I have to include that because one of the major roles of carbohydrates is to provide bulk in your digestive system, and that's provided by cellulose. Okay, now cellulose is a component of all plant cell walls, and so when we eat plants, it doesn't have a lot of caloric value. You know, some plants, okay, very low in calories. It's because, not because it doesn't have energy in it, it's just that cellulose, all that glucose in the cell walls, we do not have the enzymes to break it down. Okay, so cellulose, plant cell walls, we can't digest it, so it remains in our digestive system and creates bulk, what we call roughage or fiber in our diet. And that just creates a bulky package, right? And it stimulates the colon to contract and push the feces along. And that's because of cellulose. All right, let's talk about some lipids. Let me show you some pictures though first. And remember, all these pictures I get straight from the textbook images. So I have them under the lecture image link in the ungraded lecture resources. All right, so here's an example of a monosaccharide. So we have two monosaccharides here. We have two monosaccharides, a glucose and a fructose, simple sugars, and we add them together to form a disaccharide, which is sucrose. And then when we put a whole bunch of glucose units together, all right, we can form glycogen, which is stored energy in our body, okay, or stored glucose. All right, let's go to the next one. Let's take a look at lipids. What are the four major functions of lipids? All right, protection, insulation. There's lipids all around our body. You know, we can think about fat. It cushions our body. Help regulate physiological processes. Okay. They are a component in a lot of chemical reactions and metabolic reactions. They form membranes, which we'll talk about a little more detail, and major energy storage molecules. So we can store lipids in our body, and we can get a lot of energy, ATP, out of its metabolism. All right. Next question. What are the major functions of, actually, let's continue here. How does the structure of a phospholipid make it useful in membranes? All right, so here we go. Phospholipids. So phospholipids are just a type of lipid. Okay, this is this third function for membranes. Okay, let me see. It's a type of lipid found in major component of membranes, cell membranes. Okay, now we haven't really talked about a cell yet, that's coming up, but know that all our cells are surrounded by membranes. Okay, and now you know that a major component of all these membranes around every cell is this type of lipid called a phospholipid. I'm going to put phospholipid. All right, so it's a major component of membranes. So there are two parts to the phospholipid. Okay. Maybe I'll draw this instead. So, because the question is asking, how does a structure of a phospholipid make it useful in membrane? So this is the first part, that there's two parts to it, or two ends to it. There's a head end and a tail end, and the head end likes water. It's attracted to water, whereas the tail end does not like water. It's not attracted to water at all, okay? And because of these properties, it orients itself so that the water is on the outside and the, facing the heads, and the tails are farther away from the water. So I'm going to right now share my screen. Okay. All right. So let's take a look. I'm going to draw a phospholipid. So a phospholipid, when we draw it, is often like this. Okay, it's a molecule with different components. There's phosphates in it and carbons and hydrogens, and so we draw it just as a little circle. So there's a glycerol. Glycerol is a molecule that kind of forms this part here, and then we have these tails. So I'm going to call these the tails, and this is the head. The tails are actually made up of long strands of hydrogens and carbons and hydrogens. All right, so in other words, the tails would look like carbon, 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 and hydrogens coming off the carbons like this. All right, etc. And so we have long chains of these. So this part, the head end, is actually water loving. So we call it hydrophilic. It loves water. The tail ends hate water. They are hydrophobic. Phobia is hating something. Oh, phobic. Okay, hydrophilic, hydrophobic. Now let's dump these phospholipids. So this is a phospholipid. Let's dump the phospholipid into water and see what happens. All the head ends, so let's say here's water. All the head ends are going to face the water like that. But wait a minute, there's water over here too. So again, all the heads are going to face the water, and they just automatically line up like that. And so what we call this is a phospholipid bilayer. It's two layers of phospholipid. Okay, and it is the major component of a membrane. But picture this in 3D, right? I'm just drawing it in 2D here, 2 dimensions. But imagine it coming out into the space and forming a sphere. Okay, so it's actually going to form a ball. So everywhere around it, on the outside are all the heads of the phospholipids, and on the inside are all the heads of the phospholipid, because there's water on the inside. And all the tails are tucked away between those heads. All right. Now there's a couple other things. Inside a membrane, we have proteins and we have other things that are embedded in the membrane. And we'll talk about that when we get to the cell, but there's other molecules located inside here. And there's other things that are sticking out like this. Okay, but the majority of a component of a membrane is these phospholipids. Okay. All right, so you got to think 3D, right? Let's go back. Okay, no, actually, let's not go to that one. Okay, so this one is, let's see if I have a picture for that. Oh, I do. All right, so here's a picture of a phospholipid. like that. You might see the term used polar and non-polar. Polar means it can dissolve in water. It likes water. And then non-polar means it's not attracted to water. Okay. So this part is the non-polar part. All right. And so the head is a phosphate containing region. Okay. Let's take a look at proteins. We're almost done here. Proteins, lots of functions for proteins. All right, regulation body processes, transport system, protection, muscle contraction. It makes up structural parts of the body as well as an energy source too that can be used. Not our first energy choice, but carbohydrates are, but still can be used for energy. The building blocks of proteins are called amino acids. Okay, so we put together amino acids and to form proteins. Okay, now you have maybe heard of it, individual amino acids. People can take individual amino acids, and some must be provided in our diet. Some we have in our body. So, we're constantly breaking down proteins and building up, and so we always have amino acids floating around that can be used to build other proteins. All right, and then an enzyme. So an enzyme is a specific type of protein. And let's take a look at some things about enzymes. So this is a protein, but it's specific in that it has a particular function. Proteins or these enzymes are catalysts. So they speed up chemical reactions. All right, so we have, chemical reactions that occur. I showed you some, like let's say the bicarbonate or, building up or breaking down molecules. But with these reactions, there's usually an enzyme that is a kind of a helper. An enzyme helps a reaction to go, and it speeds it up. So without the enzyme, that reaction would take a lot more, a lot longer to happen. They're very specific and they remain unchanged and are reusable. So, and then why they important, I'm actually going to draw that also because it's easier to draw than to just verbally explain. And I'm going to bring a picture up here too. Kind of showing you what I'm going to draw too, but okay. Oops. All right. Well, maybe this is actually pretty good. All right, so the enzyme is this yellow one. All right, and so we can see it has a very specific shape. And that's what I mean by up here, it says specific. So because of its shape, it will only hold particular molecules. And molecule A and molecule B. If there's another molecule that's shaped like a triangle, okay, it doesn't fit. That's why being specific is very important. So it's very specific to the reaction that it catalyzes because of its specificity in what molecules it can hold. So once the two molecules bind to the enzyme, then it causes the reaction to occur. And you could see if these two molecules, if there was no enzyme, these molecules may never come in contact in the right angle or the right way in order for the reaction to occur. But because we have the enzyme, it's going to hold those molecules in the right way and allow the reaction to occur to get some kind of new product, a new molecule, which is then released, and that enzyme can go back and do that reaction again. So the enzymes remain unchanged and they're reusable. It can go back and pick up more molecules. And this other thing, it often ends in ASE. So if you see a molecule and it ends in ASE, it often means it's an enzyme. Not all the time, but a lot of times it is an enzyme. All right. Let's finish with nucleic acids. So nucleic acids are kind of the least, I think, known type of organic molecule. So these are molecules that form our genetic material or play a role in making proteins. All right, now the building block are called nucleotides, okay, found in DNA and RNA. So let's take a look at a nucleotide. A picture. I'm going to draw it then if I don't have a picture. Okay, so nucleotides are the building blocks of nucleic acids, and it's made up of three parts: a sugar, a phosphate, and a nitrogen-containing molecule called the base. All right, so let me draw that really quickly, because the next part is going to ask about DNA and RNA. And so remember, these nucleotides make up DNA and RNA, which are nucleic acids. Okay, so DNA and RNA are the nucleic acids. So let's take a look. I'm going to draw again. All right, so let's draw a nucleotide. Nucleotide. So I mentioned a 5-carbon sugar, and I'm going to draw it like this. So every place that there's a little crooked area where there's a bend, that's where a carbon is located. All right, so I'm going to just draw it really simply. This is a sugar. All right, here's our sugar. There's a phosphate group here. And then there's a nitrogen-containing base here. I'm going to make it a B. All right, and that is a nucleotide. Okay, that's a nucleotide. Now, there are different kinds of nucleotides. Or I should say that the phosphate and sugar are the same for all nucleotides, for all people, all right, for all living organisms. The sugar and the phosphate remain the same. How we differ is in our bases, the order of our bases. Now, in DNA, we have 4 bases: an adenine, a thymine, a guanine, and a cytosine. RNA, on the other hand, which also has a sugar and a phosphate group, RNA has its 4 bases are adenine. Instead of thymine, there's a uracil, a guanine, and a cytosine. All right, so in DNA, the sugar is called deoxyribose. Deoxyribose is the sugar. And in RNA, the sugar is called a ribose. Now, deoxy means missing an oxygen. So the sugar in DNA and RNA are the same, except in DNA, it's missing an oxygen in that sugar. But otherwise, everything else is the same, other than the uracil and the thymine ways they differ. All right. Now, DNA and RNA both are a strand made up of strands, or a strand of nucleotides. So let me draw it here while I'm here. All right, so this is what a strand of nucleotides would look like. It's no mystery, you know, DNA seems to be always so mysterious. But if we string a whole bunch of nucleotides together, okay, we get an RNA. If we have 2 strands of nucleotides, then we get a DNA. So DNA is double-stranded and RNA is a single strand. Okay, so I'm drawing an RNA here. Let me come down here. But I'll show you how we make a DNA. I mentioned DNA has two strands, but they run in opposite directions. Okay. All right. So this is tricky to draw. All right, but you can get an idea of how we can make a double strand of nucleotides running in opposite directions, and that is DNA. Okay, so a single strand is RNA, double strand is DNA. All right, let's go back. Okay, so that's nucleic acids. Let's talk about DNA and RNA. And so I kind of already started that conversation with DNA and RNA. Let me bring up all this comparing, contrasting thing. Okay, so when we compare, both are made up of nucleotides. Both are found actually in the nucleus of the cell. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So it has a ribo sugar, whereas DNA has a deoxyribose sugar. Okay. All right, I messed up my thing here. Maybe I'll move this. Anyway, all right, DNA is double-stranded, RNA is single-stranded. DNA is genetic material. RNA is important for making proteins. So it's a little different in that it plays a role in making proteins. Okay. All right. And then the bases, which I've already mentioned. So those nitrogen-containing bases, DNA has 4 bases: adenine, thymine, oh, I'm all messed up here, guanine, and cytosine. Up here. Okay, whereas RNA has adenine, uracil, cytosine, and guanine. Okay. Okay, something like that. Now for DNA, because it is a double strand, right? It's 2 strands running in opposite directions. The bases pair up in a very particular way. So adenine always pairs up with thymine, and guanine and cytosine always match up. So what does that mean? Remember when I drew that here? So if this happened to be an adenine, this would have to be a thymine. If this was a cytosine, this would have to be a guanine. So they always pair up in a very particular way, and that's what that means. Okay. Okay. What else do I want to say? Oh, I just want to add for DNA, the 2 strands run in opposite directions. All right, so remember there, and I tried to draw that. And in DNA, this is only in DNA, not RNA, because, and then these 2 strands are twisted. So this is a helix shape. So they call it a double helix, double because it's 2 strands of nucleotides, and then a helix is they're twisted. All right. All right, the last question is about ATP. I think I've mentioned it before. ATP is the energy molecule, very important. Let me show you a picture first before we get to that last question. All right, so here's DNA. We can see it's twisted here. It's 2 strands, and the purple cylinders represent the nucleotide, okay, or the sugar, the sugar and the phosphate group. So remember, the sugar and phosphate group in DNA is the same for all of us. The only way we differ is in our bases, the order of our bases. So here's the other strand running in the opposite direction. And then these are the bases. Okay, that's what I tried to draw. All right, and so that's why it looks like this ladder that's twisted. The rungs here that you step on, if it's a ladder, okay, that represents the nitrogen bases, containing bases. All right, lastly, ATP. What is it? Why is it important? ATP is an energy molecule, and let me get it the right. Okay. It stands for adenosine triphosphate. It is an adenine with 3 phosphates on it, triphosphate, and it is the main energy molecule in living things. Okay, so we make it, and the more ATP we make, the more energy we have. All right, so if you're a marathon runner or long distance runner, you want to be really efficient and good at making ATP, because then you can run farther. Okay, so here is the what an ATP looks like. It's an adenine here. Well, an adenine and it's an adenosine, okay, but it's got an adenine and it's got 3 phosphates on it. So there's something called adenosine diphosphate. So how do you get energy out of ATP? You get rid of a phosphate. So when you remove a phosphate, then it becomes adenosine diphosphate and you get energy out of it. Okay, so that's how you acquire that, or you utilize that energy. All right, phew, that's a lot of material. That was a big chapter. It's got lots of different things. So I hope you followed along and it was pretty good. And I'll see you later for chapter 3.